So I'm Cesar Costa. I also work for SPECS. So I thank the organizers for the kind invitation. And I'm going to talk about the work I'm doing together with John Listman and my supervisor Paul Verschure. Well, it was mainly his idea actually, but no, John Listman was very much involved, and I just do what I'm being told. And the basic story is about how we how we build these representational atoms in the DEC architecture, and whether we can find any evidence for how this is done in the brain. And in particular, we're going to look at the hippocampus, and I'm going to then test my model of the hippocampus on a very recent phenomenon that has been discovered in how the hippocampus is representing space. So you might remember this diagram that my supervisor showed earlier, and he also explained in great detail. And he would do that much better than I would, so maybe I will not go into the details here. But you remember that in this DEC architecture, or the SF01 architecture, we have on the one this stream of sensory states and perceptual states, as it was also just explained to my compatriot Andre, how the temporal population code would be a foundation for perception. And on the other hand, we have this stream that deals with action. And then, as Encarni explained earlier, it is sort of the integration of these representations of actions and perceptual states that I learn on the basis of correlation and prediction, as Armin just explained, which then in this integrated form forms the fundamental representational ingredient for my memory system. So now the question is there any validity to this belief? Does this make sense? I mean, on functional grounds this makes sense, and also on formal grounds this makes sense. And in the context of this DEC architecture, this looked like a reasonable proposal, but is the brain really doing this? Does the brain really integrate sensory states and actions at this very fundamental level to make that the core ingredient of memory? So this is again summarizing this, as I told you earlier. As I act in the world, I encounter stimulus events, I generate representations of these, I generate actions, and I form these couplets of sensory response elements in my memory structures. So this is now what we try to identify now, the neural substrate of this. So this is sort of the big scheme of this, and everyone tells me it's a horrible slide, but I think it's cool. So the key thing is that, so here we look at the hippocampal loop as drawn a long time ago by Ramon Cajal. And what is so special about this hippocampal loop is that when we enter from the entorhinal cortex, so this is the interface, the input structure if you want, into the hippocampus, we go from the dentate gyrus into CA3, through CA1, and then through the subiculum back again to the entorhinal cortex. We know that especially if we enter CA3, but in particular CA1, we start to see responses that are very specific to locations, as also Marty explained earlier. So, but from what are these very place-specific responses now generated? Well, if we look at the inputs to the hippocampus, on the one hand, the medial entorhinal cortex actually has a number of interesting subdivisions. And if we look at the medial parts of the entorhinal cortex, we might find cells that are so-called grid cells, and these cells will show regular responses when I'm moving through space. So that means the response of this neuron is not specific to one location. It's specific to a whole grid of locations. However, this grid has very specific and regular properties of the distance between these points and their orientation and their spacing. So, so in some sense you could think about this as giving me a very simple information of distance, but also my own actions. This tells me something about my actions in space. So the changes in these responses are due to my changes of position. These are self-induced. So if you want, this is one way to represent action. If we go to the lateral entorhinal cortex, although less is known about the lateral entorhinal cortex, what we find there are responses to sensory events in the world. And these are often fairly abstract, but they're not very specific to location. So they would roughly look like this. If this is sort of space, and here in the heat map, I indicate now the responses of a neuron in space. You might see responses to fairly high-level features of an environment, but they're fairly broadly organized in space. And we actually in the past already have built and published models of both this lateral entorhinal cortex and the medial entorhinal cortex. And the big question now is, okay, if this is sensory events, the S in my deck diagram, and these are action event. This is action because this is self-induced changes in the representation of space. How are these integrated in the dentate gyrus or in the hippocampus, or are they integrated at all? So here, that's where John Listman came in. And John Listman, who has been thinking very deeply about the hippocampus for a long time, and I was very lucky that my supervisor brought John into the project as one of our collaborators. So John Listman has been thinking already for quite a long time about how this integration of inputs from the lateral and the medial entorhinal cortex could occur in the hippocampus. And John came up with two important notions here that we elaborated. So on the one hand, it was this idea of self and non-self, right? Where medial could represent self because the changes of the grid cells or the firing of the grid cells is always due to my own movements. So this is self, implicitly action, and lateral would be non-self, the sensory world that I'm exposed to. So this is one. So this was a very useful hint for us. This is why we want to look at the hippocampus, to see if this kind of integration of now self, non-self, percept, and action could occur here. And the second thing is that John fairly recently published a really interesting paper where he looked at how inputs from the medial entorhinal cortex, from these grid cells, could give rise to place cell responses in CA3 or CA1. And the key point that John was making here is that this is largely due to predefined connectivity and not so much due to learning. And this was a really interesting and also, I believe, a revolutionary proposal, because so far the intuition had always been, well, place cells and the place field responses that they display have to be sort of learned in some way. Learning takes time. The problem is that if you measure from these neurons, that actually the place-specific responses emerge very instantaneously. It just takes you a long time to get them extracted from your data. Many passes through a certain position. So John came up with a very different concept where he said, no, it's instantaneous, just in the mapping from the medial entorhinal cortex onto CA3 or the dentate, this place-specific response is generated automatically. And now what I looked at in my project, my PhD project, is how now the projections from the lateral entorhinal cortex contribute to this mapping.